Speaking of dark, this is what you missed, or you missed me as the case may be, last Wednesday. So OMSI After Dark, it's a lot of fun to head over there, and Stanley the SSV was obviously having fun as well when they, something or other was over there at the time. So it was really a fun time to chat with various different people. Turns out one of the people who came by my demo had worked on bacteriophage T4 at University of Illinois at the same time that some of my real heroes, people who discovered the archaea, Carl Woese and Ralph Wolf, were there. And then a couple of alumni, or I should say alumna, from this class came by and said, I'm so glad you're not going to ask us T numbers for all of your models that you brought today. So that being said, let's get started with finishing up a little bit with Lambda, and then moving on to plant viruses. And the plant viruses is just one chapter in our textbook. Unfortunately, it's in the new edition of the textbook. It's one of the few that is not in the current version of the textbook, or I say the first version of the textbook. And how did I get so far ahead? The second edition is on reserve in the library. So if you need to go and check it out, you can do that for a couple hours over there, just particularly for this chapter. And I may have a PDF. I forget if I scanned that and posted it to D2L. If I haven't, I will try and do that a little bit later. So we'll finally get to talk about these guys today, and then, as I mentioned, a little bit more about Lambda. Last time we finished up right before we got to talk about integration, potentially one of the most fascinating aspects of Lambda biology, and this has to do with how you get the Lambda genome, shown here in brown, into the host genome here in black. And it's about the attachment site. So the attachment site in the phage at P, and the attachment site in the bacterial genome at B. So this is also often shown as the phage sequence on one side, the operator site, and then the phage sequence on the other side, POP prime, and then equivalently in the bacterium, BOB prime. As I mentioned last time, but it bears repeating, Lambda integration is a site-specific integration event. So it only goes into one place in the genome. And that's because these sequences, particularly this O sequence, is repeated in both the at P and the at B part of the genome. And then these other sequences, the flanking sequences, P and P prime and B prime, are bound by the integrase protein. So the integrase protein, again, only expressed in lysogenic growth, binds to the arm. So what the heck is an arm? The arm is also just another name for these flanking sequences. You also have host proteins that are involved. This, again, ridiculously creatively named IHF. IHF stands for integration host factor, again, because people discovered these when they were looking at Lambda. Turns out that integration host factor is a really important gene for compacting the genome of E. coli. So bending. That's one of the things that was very early discovered, that it bent DNA. And it turns out this bending of DNA is really critical for getting this integration event to take place. So integration together with integrase and the integration host factor that takes the viral genome and puts it into the host. And in that process, you end up with bacterial sequence over here and bacterial sequence over here, with the bacteriophage sequence in between, and then this repeated sequence, the operator sequence, in between those. So that's going in. That's the lysogenic state. And somebody asked me a question that I posted on D2L. So D2L activities, discussions, questions. It's kind of a hard place to find them. But any questions that people ask me via email, or usually it's an email question, I will also post in that question section. So if you have a question, you're interested in potential answer to it, go and check there as well. But the question here was whether integration only takes place during this lysogenic growth. Yes, it does, and that's because of the regulation of the integrase gene. So once you have this integration that takes place, this is great and wonderful, but we also have to have induction. And so when the going gets bad, C1 gets cleaved, and then the virus can come back out. How does it come back out? If it's stuck in the genome, there's no way this can actually be replicated and packaged into normal virions. So it has to get cut back out. And it turns out that the integrase protein and IHF are critical for this. But then you also need this excisionase, XIS factor. And so this is what drives the reaction here to excision. In the absence of excisionase, you're going to end up getting integration. And that, again, not surprisingly, the expression of this integrase, I'm sorry, not the integrase gene, the excisionase gene is repressed by the presence of what protein? Who's up next? We're almost done with the list, and then I can start again. Oh, yay, oh, joy, oh, rapture. Aspen, Lindsay? Aspen here? Cheyenne? What proteins? We get to the end of the list, and then I'm going to have to ask for volunteers. It'll be frightening. Rosalind? No? Man. It's just, it must be too hot in this room. Adam, it is ridiculously hot in this room. I'm sorry about that. I can't do anything about it. Adam? Yeah. Yes. What do you think might be regulating the excisionase protein, repressing it, for instance?
The abundance of the proteins that either suppress glycogen or encourage it.
Mm-hmm. So what is going to encourage lysogeny? Yeah, so, well, C1 is going to be helping keep the lysogenic state. So in the absence of C1, which just happens with induction, so the DNA damage that gets cleaved, so the excisionase protein is actually repressed by C1. And so it will be expressed, and it turns out it's expressed as part of the, remember correctly, that would be PL in the virus genome. So, but it makes sense that this is why you get the excision now of the genome. So once it's excised, or if you're just going through the normal lytic process, there are all of these alphabet soup proteins, which reminds me, I'll be having office hours here today, right after class, instead of heading back over to my office. And I have the DVD that I didn't show last time. We can have special viewing of that here after class if people have some time. And then I will be going to put it on reserve in the library. So you can go and check it out from the library as well. So the O protein binds to the origin of replication, so serves very similarly to the bacterial DNA A protein. Then the P protein recruits DNA B. Okay, we've got two more to go through here. Mani, what do you think about the DNA B protein? What does that do? It's a binding protein, not particularly a binding protein. What's the most critical step in terms of starting replication? What do you need to do? DNA replication. How's that? Right, exactly. What is that? Pardon? Helicase, exactly. So that's the replicative helicase. The DNA B is a replicative helicase. So you bind to the origin, you get the helicase. Once you have the helicase, those two strands are separated, so you will have DNA replication that will take place because the DNA primase, et cetera, will come down and bind. The early replication that happens on the Lambda genome, remember it's a circular genome, happens just like E. coli replication does. Two replication forks facing away from each other. They replicate around the genome. Those polymerase, the replication forks, bang into each other. Magic happens, and you end up with two genomes. Actually, not very well understood how termination happens. However, what happens in terms of making more genomes, we all remember, because we've run out of names to call here, that it's packaged as a linear piece of DNA. And as we've talked about before, these are concatemers. And this I actually like as a nice other way of talking about concatemers. The way you make concatemers is through this rolling circle replication, which I wanted to take a look at here because I think it's a nice description of how rolling circle replication actually happens.
By conjugation and reproduction of certain viruses,
which I don't. Okay, I don't like that. So you have a nicking enzyme, and what do nicking enzymes do? They just cut one strand of your DNA, generating a 3'OH. What do 3'OHs do? They serve as a primer for your replication. And so replication is happening here, and then helicase activity is separating this second strand. Then, if you've got a single-stranded piece of DNA, just like you would have with the DNA helicases, now you have DNA primase, which will make a second strand. RNase H will get rid of this, and you end up with multiple copies all hooked up end-to-end just because you're replicating around and around and around in this process. So if we go back to here, this is what I'm trying to demonstrate here with this circle, and then many, many, many copies of the genome that come off of that. And so that's shown here in the textbook. This is that concatemer. So there should be, hopefully relatively obvious at this end, there's kind of a cutoff, and then you have a cohesive end site, another cohesive end site, and of course another cohesive end site, another cohesive end site, etc. So that's your genome during late replication. Again, stretched out as this concatemer with all of these COS sites in it. So the next thing that happens after you have this, again, multiple copies of your genome and concatemers all hooked up to each other, then you have the terminase protein, which binds to one of these COS sites. And again, COS stands for cohesive ends. This is where they're going to hook up together when you have infection later on to make a circular genome. That binds and cuts at this COS site, and then it goes and finds a prohead structure. So we talked about procapsids before. This is just part of the capsid because the whole thing would include a tail. The prohead now has the terminase attached to it, and then through large amounts of hydrolysis of ATP, this gets stuffed into this head structure, shown here as this coil, until this next COS site gets to the terminase, at which point the terminase cuts, and you have a head now filled with DNA. This head is made through scaffolding process, has chainmail, which is these cross-linked side chains that are making peptide bonds in it, and then there's this weird protein that was originally discovered because it was necessary for assembling Lambda. Why else do we know about GRO-E, or GRO-EL, GRO-ES? Margot in the back. Chaperones, exactly. So chaperones were discovered because of the study of bacteriophage. So this is where, again, why bacteriophage are so awesome. So chainmail, we already talked about this terminase protein. It also goes through a portal very similar to what you see with bacteriophage T7. And now you have this head with the DNA in it, but there's no way that this is actually infectious. You have to add this tail. And one of the really fascinating things about the tail of Lambda, which was discovered by this guy, Roger Hendricks, also with a favorite virus of mine, in Steamboat Springs, Colorado 2 years ago. What he discovered is there's a protein in the bacteriophage Lambda genome which literally measures how long this tail should be. So what it does, it's a protein that's got a certain length. If you shorten the protein, you have a shorter tail. You lengthen the protein, you get a longer tail. And as I mentioned before, last time, that these are actually all donuts. This is not a helix. They're literally donuts which are stacked on top of each other, and just stacked in the process whereby you have all of these lined up at the appropriate length. So now we have virions. They need to get outside of the cell. How do they get outside the cell? Again, this is E. coli, so you need to get through the inner membrane and the outer membrane. So how do you do that? You've got peptidases, endopeptidases. What would these be cleaving? The protein part of the thing that's in the periplasm, peptidoglycan. So that's what these endopeptidases are doing, and then Holins to get that endopeptidase into the periplasmic space in order for it to be broken down. A number of people have also asked me about the regulation process. This is all you need to know about lambda regulation. This is a figure actually from that book that I brought last time. It's all about C1 and CRO and their regulation of N. I'm not going to go through this whole process, but I find it's actually kind of a nice review to think about how lysis and lysogeny are being driven. And again, this is int and that process, pardon me, int here and C1 are what you see in lysogeny. And unfortunately, the colors didn't come across here very well. But this is supposed to be orange versus red. Orange is now going to be your lytic cycle. So again, where did lambda come from? It was a mistake. The structure with this cylindrical tail as opposed to a helical tail, but otherwise a pretty normal head structure, binds to Lambda, which is a maltose transporter. I probably should have put this in red. The big story here is about transcriptional regulation. Via lytic growth, which is we had our clicker question last time, is about anti-termination. Lysogenic growth, which is all about C1, which you can see all these nice clear plaques here because this is one of those C mutants. I believe this is a C1 mutant. And then the choice between lytic and lysogenic growth, which has to do with the growth state of the cell and the activity of C3, which hopefully makes sense when you look at this when you're reviewing in like weeks and weeks when you've already finished your other midterms and have to go back and think about this class. And we just talked about replication. Starts out theta replication, then goes to rolling circle, how it gets out, and then, as I mentioned before, a DVD. So questions on lambda? I'm sure you have absolutely none. Yes, Nicole?
The prohead, is it only considered a prohead until the genome is put inside, or only until the tail is hatched?
Okay, so the question is basically when do you consider it a prohead versus a head? Just again, to paraphrase. So first thing, again, a lot like what we talked about in terms of capsids and virions. First, you have to get the genome in, but then you also have a conformational change, which is this cross-linking that happens. And so you may remember it was sort of shown as a circle, and then it goes to an icosahedron. And that is that process going from the prohead to the head. You also lose the scaffolding proteins in that process. Yeah?
What is it that's actually stuffing the DNA into the capsid there?
Oh, so what is the protein that's stuffing the DNA in? In this case, it seems to be a combination of the terminase and the portal protein. We talked about the portal protein as being the really important one for T7, but here's the terminase and the portal protein, which seem to be important. Yeah?
It's just internal scaffolding.
Again, just internal scaffolding. The only one that's got the external is that FIX174. In fact, the only one we're going to talk about that has external scaffolding.
So what causes the conformational change from the round to the, to simplify it, from round to icosahedron? Is the internal structural proteins?
So again, the question is, how do you go from circle circle to icosahedron, or sphere to icosahedron, I should say. It's a whole combination of things. It's putting in the DNA, it's removing the scaffolding proteins, and getting all those cross-links. So all of those multiple things happening. Yeah?
Are external scaffolding proteins usually more rare, or are they just not being recognized?
Oh, yes. So the question is how common, again, I'll just paraphrase, how common are external scaffolding proteins? They're actually really pretty rare. It just seems to be present for those microviruses. So you have, it's pretty rare that you have these external scaffolding proteins. There may be some other ones, but I don't know of them. And as I was mentioning before class, I'm not completely omniscient, so there probably are some other ones. But as far as I know, it's just the microviruses. Yeah? How does the tail get attached? Good question. There are other proteins here, right at this junction. And it's in the DVD, but it won't be on the exam. That interaction doesn't seem to be recruitment. It just seems to be stochastic. Most of the things that happen with virus assemblies seem to be very stochastic. So even this interaction, for instance, with the terminase and the portal protein, they just seem to be floating around. And it depends on your concentration. As long as you have a high enough concentration of proheads, high enough concentration of genomes with terminase, they will interact with each other, and that then will be a positive interaction, at least as far as the virus is concerned. Okay, we're all so happy with these guys, we can answer a clicker question, right? Yeah. I love that enthusiasm. So, again, let's get started on here. Go. Are we going to go or are we not going to go? Yes. Okay. So we have a minute. Again, make your decisions. I will see what decisions you've made. Nobody's made any decisions yet. We got one vote. Oh, that would help. I just guess. So that's the first round, right?
Come on, it's not a midterm.
No one will guess on my midterms. So I would say late genome replication here. The majority of genome replication. That'd be a great question to ask me. So there's a question like this in an exam. Dr. Stedman, do you mean early or late? Okay, 36 of you voted. Now tell each other what you voted and why. Pardon? You can vote. We're going to give it a chance. You just discuss. Just say what you would have voted if you had a chance. Okay, we're getting quieter, which means that everybody has decided. No, you haven't? You're cogitating. You're passing out because it's too warm in this room. Okay, let's try this again.
If we don't repress, does it just give the old answer? Do we have to repress?
You do need to re-vote, yes. And you can vote many times. It's the last one that you press that counts. So much fun to see what your answers are up here. And there are more of you voting this time, which is good. Anyone else want to vote? Got 10 more seconds? Your thumb's going?
How many people are in this class?
Number of people who took the exam, which I think is probably a good indication. I think it was 67? So yeah, so there are a few people who aren't here today. Okay, so it's actually good that you talk to each other because there was a much more of a difference of opinion earlier on. So let's take a look. We're not 100% yet, but we're getting close. So why phi x 174?
Rolling circle replication.
Rolling circle. That's what it's about. So, and the key here really has to do with the mechanism of that late, and again I should have late here, genome replication. So that mechanism is the rolling circle mechanism. Okay, so that's it for Lambda. Now let's do total injustice to plant viruses. Because it's one of those subjects. And in fact, if you go to OSU, you can literally do a degree in plant viruses. And I think they've got 3 quarters worth of plant virus courses. So we'll just very briefly touch on this. And for people who got here a little late, this is a chapter which is not in the first edition of the textbook, but it's in the second edition, and that is on reserve in the library. And I will try and post a PDF copy of that as well. So big picture here is that most of these plant viruses are RNA viruses. There are definitely some DNA viruses that infect plants as well. And many of these are at least the ones we're going to talk about today, are the so-called Mosaic viruses. And the way that a lot of these plant viruses are named, they're named based on their host, followed by what the phenotype of disease is. So Mosaic virus, and we'll see CucumberMosaic virus. So many of these are named in this way. One of the big differences that a lot of these plant viruses have, particularly the small RNA plant viruses, is they have segmented genomes. So we haven't really talked about segmented genomes before. Just very briefly, sort of like to think of them again. It's kind of like individual chromosomes. And then because they're getting into plants, they also need to move from cell to cell. And that's one of the big areas of research in plant viruses, is thinking about how they move from cell to cell. At least in humans, once you have a virus that gets into your bloodstream, that's clearly circulating. Plants have very tough cell walls, and so getting into the plant cell in the first place and going from cell to cell turns out to be a real challenge. And so these viruses have developed ways of doing that, and we're probably not going to get to tobacco mosaic virus today, even though it's totally awesome and wonderful. But again, I could spend a whole course talking about this. So a couple of key concepts. We talked about RNA viruses before. We talked about the RNA phage. Basically, from here for the next 2 or 3 weeks, we're going to be talking about RNA viruses. So some of those key aspects of RNA viruses, transcription, or I should say replication versus translation, is always going to be a bit of a problem, and positive strand, negative strand, et cetera. Again, these segmented genomes. Conformational changes in terms of getting into the genome, getting inside the cell, some of the host effects, and then hopefully we get a chance to talk about this really cool heat resistance story. Actually, it's not a plant virus, it's a fungal virus. And then TMV is the classic helical virus structure. So the subject of this particular chapter, again in the version 2 of the textbook, is CucumberMosaic virus, or the Cucumo viruses. Again, the naming things here are really kind of crazy. But we'll talk a little bit about where it came from, the structure, entry and transmission, replication, and packaging. And then, depending on where we get to, we'll talk about some of these guys. This should be really clearly obvious exactly what kind of T-number structure this is. T equals 3, but we'll get back to that in just a second. Reminder, these are, again, this particular set, I should say, CucumberMosaic virus is a positive strand RNA virus and single-stranded. So this can be translated directly, and particularly important is that these guys are now going to make all of your early proteins. That particular RNA, the RNA which is released into the cell, that's going to make your early proteins. The late proteins are going to get made from newly made genomes because you need a lot more of those late proteins. And so we'll talk more about that later. So you have to have genome replication before you're going to get production of a whole bunch of these late proteins. Where does CucumberMosaic virus come from? It's a virus that was discovered actually quite early in the discovery of viruses, 1916, right about the same time that the bacteriophages were being discovered. These actually kind of look like some of the cucumbers I see at the farmer's market. This is a non-infected, but then when you start to see this discoloration and some of these extra growths here on the cucumber, this is a pretty typical phenotype of this CucumberMosaic virus. Turns out it actually happens as leaves as well. It's not just the fruits that are happening. And then they push this CucumberMosaic viruses as having very wide host ranges. Oh, well, okay, Stedman puts it in quotes. Why would he be putting it in quotes?
Not true.
Well, it depends on your definition of what a very wide host range is. So if you look at molecular diversity by the small 7 RNA sequences, all plants are this tiny little insignificant twig down here next to the insignificant twig of humans, etc. So molecularly, they're not terribly diverse, but they do infect lots and lots of plant species. And this particular, the CucumberMosaic viruses, actually infect about 1,200 species of plants. So we usually talk about viruses having pretty narrow host ranges. as far as numbers of species, these definitely do have a reasonably wide host range. What does the structure look like? This should be really, hopefully, obvious that this is a T equals 3 structure. I hope it is. So we've got a 5-fold axis of symmetry here, a 5-fold axis of symmetry here, a nice 3-fold axis of symmetry, and a hexagon right here. So you go H of 1, change direction, K of 1 gives you a T equals 3 virion, and I brought up a whole bunch more of my actually plant virion, 3D printed virions up here at the front. What makes that structure? It's a beta barrel structure, but instead of the double beta barrel structure, which we've seen for a number of different viruses, and some people, including me, think is evidence for really ancient viruses, those beta barrels actually stick up perpendicular to the surface of the virion. These guys are flat as far as the surface of the virion. And we'll hear more about these structures a little bit later on. So you have this beta barrel, which is really forming the surface, and then you have this arginine-rich N-terminal alpha helix. What would arginine-rich alpha helices like to interact with? Acidic things like? So what's going to be on the inside of a virion? A genome, in this case the RNA. So that interaction. Turns out that's actually a misprint in the textbook. The textbook actually says that there's a highly negatively charged part of the protein which is sticking on the inside. No, that's incorrect. So don't always believe what you read in the textbooks. So hopefully the 3rd edition will have corrected that problem. I actually emailed them about that. So, so this is the virion. Now how does that virion get into the really tough plant cells? And this is actually probably why these particular viruses have such a wide host range, is that they can't get inside the host cells by themselves. They need these guys. So what happens, the way you can actually get inside a plant cell is to be brought in with something else that is degrading that plant cell. And so these viruses seems to have evolved together with these plant-eating insects. And this particular one, aphids, transmit many of these Mosaic virus-like and also particularly CucumberMosaic virus. So what happens here is you have, pardon me, the aphid poking its mandibles into the plant and thereby transmitting all of these viruses. Yeah?
Does it do anything, is it just structural damage? Like, do you even know if you eat the cucumber, or does it completely ruin the plant?
Oh, the actual phenotype of the disease. So does the disease actually ruin the plant? These, again, it really seems to depend how much of the virus you happen to have, and then also the plant defense mechanisms against those viruses. But some of these, again, I see these things at the farmer's market, and I would say, okay, maybe it's got CucumberMosaic virus associated with it, but it seems to be okay. So it just seems to vary. But you also will have some phenotypes on shoots and leaves, and those then you get much, much, much lower yields. So yeah, but what I thought you were going to ask was, is there something specific about the aphids bringing in that particular virus? And does it have to pass through the aphids? We'll see this is true for a lot of the human viruses later on, but actually no, it's just literally plant damage and damage to the cell. So when researchers work with these plant viruses, usually what they do is take a little bit of sandpaper and sandpaper the leaves and put the virions on it. So it is literally just the damage which seems to then allow this infection to take place. Yeah?
Is it the damage by them inserting into the plant so then the virus can go in, or are they actually carriers of it on their person?
Yeah, their person. No. Yeah, so the basic question is, I'll paraphrase here, is where are the virions? Yeah. And the virions just seem to be on the outside of the mouth parts. Okay. So nothing inside. They're not getting the salivary glands, all that other extra stuff that, again, we'll talk about later. Yeah, no, they don't. They're just, so it's literally a guilt by association. It's sticky, and those end up inside. Yeah, Jacob. Why not why not use agrobacterium to get things in? It's so much easier to use sandpaper than to grow a culture of agrobacterium. And agrobacterium is also much more specific in terms of transmitting its DNA. So that's, yeah, sandpaper works, just scratching it a little bit. You know, any kind of just abrasion of the plant leaf surface, you can get infection to take place. And it's literally just cell damage. That's all you need. A little bit of cell damage. And then once the virions get inside those cells, as we'll see, they can then move throughout the rest of the plant. Yeah? So for CucumberMosaic, the question is, are aphids the only vector for these plant viruses? For CucumberMosaic virus, it seems to be just aphids. But for a number of other ones, you have other insects which seem to be the vector for moving these particular viruses around. And so as I mentioned before, these guys have these segmented genomes. But what's really fascinating about a lot of these different plant viruses is that they need multiple virions in terms of being able to get a productive infection. Because they need all of these different genome segments, and it turns out those genome segments are actually put in separate capsids. Just very, very bizarre the way that this seems to work. And let's just take a quick look at that. So if you look at CucumberMosaic virus, an infectious unit is actually going to require these 3 genome segments, and actually there are 3 major genome segments, and again, ridiculously creatively named RNA1, RNA2, and RNA3. These RNAs, again, we'll talk about how they replicate in a couple of minutes, but each of these gets put into one capsid. So if you have one capsid, it's going to have RNA1 in it, a different capsid is going to have RNA2 in it, and yet a third capsid is going to have RNA3, and actually often RNA3 and RNA4. So do you notice anything particular about these different RNAs, RNA1 and 2 and RNA3 plus 4? They've all got funky structures on them. We'll talk about those funky structures in just a minute. What else? They make different proteins, exactly. You need those proteins for replication purposes. What else? Variable lengths, but key should be down here. These guys go together into one virion.
Oh, the capsid can only hold like 3,000 something base pairs.
Exactly. The capsids themselves, that T equals 3 capsid, can only package about 3,000 nucleotides of RNA. So that's probably why these things have evolved to have this kind of multiple segments, each of them, 3,000, be nice small capsids, but you need 3 of them. So what does that mean about this infection process on the aphids? So the aphids would have to carry all of these individual capsids and all 3 of them together. And again, if we think about the statistics, it's not going to be all 3 all the time. Sometimes you have 2 of the first one and 3 of one of the other ones. So what it means is anytime you have an inoculum, you've got to have a whole bunch of capsids coming in at the same time. Now, this is a micrograph, SEM, of looking at an aphid. But relative to the size of the virions, this thing is ridiculously ginormous. So you have literally millions of capsids on the mouthparts of any one of these plant-eating insects. So statistically, you will have all of the capsids that you need in order to get a productive infection. Yeah?
So what are you looking at in terms of MOI? You need 10 before you get big and start turning the cell. Is this like one of the exceptions?
Right. So the question basically is, what are you thinking about in terms of MOIs? Because all of those MOI calculations are based on infectious unit. Now you've got all of these extra capsids that you need in order to get that infectious unit. And so one of the things that's mentioned in a previous textbook is having multiple hit kinetics. So you actually would have to have, and this is why you can use your Poisson distribution, you say you have to have at least 3 in order to get. So your P of 3 now, how many, what does your MOI have to be in order that you have to have at least 3? And then you also have to hope that those 3 are the correct ones, so you can do even more calculations to figure out how much. So what that means is you have to have a way higher MOI to get really productive infections. And the way that that can happen is because there's huge numbers of virus particles that are associated with the aphid mouthparts. Now, why are there so many virus particles associated with aphid mouthparts? When you look at diseased plants, it turns out that they make ridiculous numbers of virions. And so if it's a uncontaminated aphid, as soon as it eats a contaminated plant, it's going to get, again, literally millions of virions that are associated with the mouthparts. And as soon as it goes to another plant, those millions are going to end up coming back in. So the MOI in this case ends up being so high that it's not usually a problem. But again, it's because of this infection mechanism, which seems to evolve because you have to get in through this tough cell wall. Isn't evolution amazing? Yeah. So that being said, in each of these RNAs, we have a couple of different things. Again, these are RNA viruses, so they need RNA-dependent RNA polymerases. There's no cellular RNA-dependent RNA polymerase. That's to make more genomes that you end up kind of have to have for your late proteins. The late proteins are encoded on RNAs 3 and 4. This is the capsid protein here. And then RNA3 also has a movement protein, as we mentioned, the movement protein. So it's great if you have an infection on the leaf, just where that particular aphid happens to have gone in. But if that's the only cell or set of cells that those viruses have gotten into, you're not going to have a great infection. So these viruses, once they get inside the plants, then they need to get around inside the plants. And then you also have this really fascinating multipartite protein here, encoded by RNA1, which has a methyltransferase and an RNA helicase activity. Methyltransferase. Why would you want to have a methyltransferase? Prevent exonucleases from going at it. And what's a very important part of this structure right here? The cap. Cap has methyl groups on it. So the methylation of the cap comes from this methyltransferase. Why do you need methylated caps? To protect, but also what do you need it for? I've heard mutterings. So RNA processing to some extent, but what particular kind of RNA processing? What do all viruses need? Every single one. We haven't found any virus genome yet that encodes a ribosome. So translation. It's all about translation. How do you get translation initiation? You have cap binding proteins that associate with the cap. You have an RNA which has come in from a virion. If you want to get it translated, you need to have a cap. How do you put a cap on one of these things? Normally, capping happens where? In a non-virus infected cell. Where's capping happening? In the nucleus. Where are these guys replicating? Cytoplasm. So translation, everything's happening in the cytoplasm here. So this cap has to come from somewhere. It comes from this methyltransferase. And we'll see why we need this RNA helicase a little bit later on. Okay. Any more questions? You're all going to be able to answer the next clicker one? Easily? Yes? We'll all get it right? We'll get 100%? Let's do it. And you can see it now. Sorry for last time. Oh, let's start. That would help. And if everybody gets it right, we don't have to have discussions. So you can vote for something else so we can delay everything. All kinds of ways to game the system. I was told if it's more than 80% correct, I should just not bother. So.
The right get righter and the wrong just get left behind.
That's your 20%. The wrong getting wronger. More than 90%
Oh boy.
are getting it right. So I suppose we won't have these long discussions because everyone knows already. Yeah.
So what is the determinant of that additional fourth variant that's required?
So,
What designates that you need that additional RNA4?
okay, RNA4 is being packaged with RNA3.
But not
So-
always. You said it's not always there.
Did I say it was not always there?
You said it's not always there. Sometimes it's included, unless I misheard.
Okay. Pardon? Oh, and often RNA4? I should have said to be packaged appropriately, it always does get packaged together. These are subgenomic RNAs, which we'll talk much more about later when we talk about other viruses. So pardon if I misspoke on that. That would not have surprised me. Again, I'm neither omniscient nor infallible. So it's good to call me on these things. Okay, so that's the different parts of the genome. You may have seen that funky-looking clover-leaf structure at the 3' ends. If you zoom in and look at that in a little bit more detail, it gets even more interesting. So we just talked about the 5' ends again, methyltransferases. That's what's happening there. 3' ends. Usually, what happens usually at the 3' end of your messenger RNA? What's there? Polytail. Exactly. We don't have that here. So we have this structure. And very often, now it's not going to be this exact structure, and I'm not going to expect you to remember the nucleotides that are here, but this structure may not look like it, but it's actually very similar to a transfer RNA. So transfer RNAs are great for a lot of different reasons. One of them, they have very stable structures. And so that stable structure is going to be at the 3' end of actually each of those individual genome segments. So why would you want to have this? What is one of the things that poly A tails do? And we talked about this, what caps are for too, right? Protecting the ends so they don't get degraded. What do the hungry things in the cell like to do? Chew in at ends. So the 3' end right here, just of the genome, is CCA. What happens in a tRNA with the 3' end CCA? tRNAs. Amino acyl tRNA synthetases. What do? Add an amino acid to them. And it turns out that cellular amino acyl tRNA synthetases do this to the end of plant genome sequences. It's really pretty amazing. So most of these plant genomes are going to have a, amino acid actually attached to this 3' end of the genome. Varies from plant virus to plant virus. In the case of CucumberMosaic virus, actually tyrosine, if I remember correctly. Maybe it's tryptophan. But gets added to the end of the genome, and then also clearly protects it. Also, these seem to be important for not just protection of this one amino acid. What else are tRNAs used for? Translation, exactly. So translation, get translation to happen, you have interactions of the tRNAs with the ribosome and all of the initiation factors in order to get translation to take place. So how do you get the translation factors associated with your viral RNA? You interact with this tRNA-like structure. So serves to protect the ends, serves to also help with translation procedures. So this, I notice, is something that I basically talked about already. RNA1, methyltransferase, important for 5' capping of ends. RNA2, your RNA-dependent RNA polymerase. RNA4 is the capsid protein. What we haven't talked about are these viral proteins 2B and 3A. I can't remember what all of these are, so I wouldn't say don't bother thinking about what each of them are. Think about their activity. So again, methyltransferase, important for making ends. RNA-dependent RNA polymerase for making more of the RNA. And capsid, of course, for making capsid proteins. These guys, 4A, RNA, 2B, and 3A, you see I keep forgetting them as well. These are the ones which are important for movement and movement inside the cell that we'll talk about in just a second. But first I wanted to mention replication. Replication, so back up here. This viral protein 1A has methyltransferase domain, but also has an RNA helicase domain. And that RNA helicase domain seems to be really important for the replication taking place in these cells. Now, One of the things that we haven't talked about too much yet, but we'll continue to talk about more and more for the rest of the RNA viruses, is that double-stranded RNA inside a cell is really, really bad as far as the virus is concerned. Because anytime you have double-stranded RNA inside a cell, what does it mean to the cell? You've got an RNA, and particularly an RNA which is being replicated into RNA. What does that mean it probably is? It's probably a virus. And so cells have developed really good systems for detecting double-stranded RNA. So all of these RNA viruses that have to make that negative strand are going to be trying to protect themselves from getting degraded. So RNA interference. There are other proteins as well. We'll talk about translational inhibition that happens with double-stranded RNA. So basically, the virus wants to protect its RNA from the rest of the cell so the cell doesn't see that there's some kind of virus replication going on. The way that this works in CucumberMosaic virus is by making an extra compartment inside the cell in the endoplasmic reticulum. And we'll see this happens again lots of times with these RNA viruses. There are extra compartments that are made through interactions of virus proteins. So this virus protein 1A, which also, again, is the methylase and seems to be important for making the, say, the RNA helicase and separating these different things, also will form one of these sort of membrane compartments inside the cell and interacts with a secondary structure at the 5' end of the genome. So again, when you first release the genome inside the cell, this is going to get translated. After you have translation, you're now going to start making these viral proteins. Eventually, you make enough of these viral proteins that they will start to bind to the RNA. As soon as you bind here at the 5' end of your RNA, what does this do to translation? Stops translation. Why is stopping translation so important?
It keeps the cells protected.
That's one thing. It keeps the cells protections. What else? What's the big problem with RNA viruses? You translate and you replicate. So they run into each other. If you stop translation, now you can replicate. You don't have your RNA-dependent RNA polymerase running into the ribosome all the time. So, and how do you control this? Well, these proteins aren't made until after you've done some translation. And then they start to bind. And then you also will make these compartments. And then in these compartments, now you will make more of these single-stranded RNAs, which will end up being packaged, et cetera, in order to finalize the process. There also are lots of host RNA binding proteins that are important. Many of those, not surprisingly, interact with these tRNAs and their translation proteins. Again, just like we talked about in terms of the RNA phage. Translation proteins, particularly the initiation factors for translation. Eukaryotic EIF1A interact with these proteins. And again, they're RNA binding proteins that are helping to get translation, but are also used to help with the replication complex. The other thing we haven't talked about as far as these plant viruses are these movement proteins. What do movement proteins do? Particularly this protein 3A from CucumberMosaic virus. It targets the RNA to these plasmodesmata. Plasmodesmata, excuse me. And I'm not a plant virologist. I would never have claimed to be one. But these plasmodesmata are these connections between various different cells, highly regulated what gets passed between the different plant cells. This protein, once it's bound to the individual RNAs, will bring them to plasmodesmata. Those will then get transferred from cell to cell to cell and eventually end up in the plant vascular system. So when you have the aphid that takes a meal here, will release these virus particles. They'll end up here in the epidermis. Then through the plasmodesmata, will eventually end up at the plant vascular system, xylem and phloem. This will now move these viruses throughout the whole rest of the plant. So you do literally just need to take one leaf, scratch it a little bit with your sandpaper, put these virions on it, and they will spread throughout the whole rest of the plant. There's all kinds of fascinating plant defense mechanisms that try and deal with this that are way beyond the scope of this course. So that's CucumberMosaic virus. I want to spend the last 10 minutes or so talking about some really cool other things that happen with plant virus. Oh, Margot, yeah, before I move on. So the question is basically, how do you get out of the vascular system and into new cells in the rest of the organism? The easy answer is I don't know. But the other one is there are certainly also plasmodesmata in these connection systems through the rest of the plant as well. And it's probably these movement proteins in complex with the RNAs, which are then being moved. So it's really a complex, and I should have here 3A, back up real quickly. So 3A plus genome segments here. And so it's that complex which seems to be moved around. They do seem to stay with it. So the movement proteins seem to stay with the RNAs as they're moving through. Okay, so what's this funky image that I keep showing in the beginning of all of my lectures? This is CCMV, cowpea chlorotic mottle virus. Again, one of these, what's the host? What does it cause? Don't ask me what a chlorotic model is. I don't know. But these are really fascinating because they have this wonderful structure. Again, T equals 3, 5-fold axis, 5-fold axis, step one, step the other. But what's really nice about these is you can look at the conformational change really easily just by changing pH or changing ionic strength. And so these guys just literally open, but they can also close. And so if you change the pH, they open. You can change the pH back, they'll close, open, close, open, close, open, close. Turns out they're really easy to make. You can make them in plants by just scraping the leaves and putting the virus on them. But you can also just express that coat protein, and they will make all of these capsids. And so one of the neat things that you can do is make this capsid. Again, it's not a virion because it doesn't have a genome associated with it. And then treat it with different pHs, different ionic strengths, and it opens and closes. Why is this so useful? Well, it turns out you can package all kinds of different things inside CCMV or any of these other ones. So this is an example of all of these capsids lined up right next to each other that people have put various different metals in, and they're particularly interested in ferromagnetic metals. So you can actually use these for very high density storage. In fact, the group down here, Mark Young and Trevor Douglas at Montana State, now in Indiana, they were working with Panasonic to develop really, really high density hard drives, because these particles are very small, smaller than it's easy to make metals in any other way. So what you do is you open up the particles, put in some of these ferromagnetic metals, let it close, lay them all out next to each other, and then just get rid of all the protein, and you've got all of these metals lined up really nicely right next to each other. Really fascinating process. Also potentially important for MRI, contrast agents, again, adding a lot of these metals. You can also think of applications where you use these for chemotherapy drugs. Again, you put the chemotherapy drug on the inside of the capsid. You get it to the cancer cell, somehow convince it then to open. You can release the chemotherapy drug. So really neat biotech applications of understanding virus structures and virus entry procedures. So that's just one example. There are lots of other examples that people are using, although this is one of the best discovered ones, or the best developed ones, excuse me. And full disclosure, I did a postdoc with Mark Young at Montana State, so I like the work that they're doing there. Speaking of Montana State, this is a connection to something that I heard about while I was there. This was in, 2000, 2001. As many of you know, I poke around hot springs and try and find the viruses that are in the hot springs. One of the things that you notice whenever you're in one of these hot spring environments, so the next time you're in Yellowstone or Lassen Volcanic National Park, etc., if you see this plant here, Dicanthelium lanuginosum, also known as panic grass, these are really amazing plants because their roots can grow up to over 50 degrees Celsius. And literally, just by going out into one of these hot spring environments, just looking where these guys are growing, you can tell what temperature the ground is there. So 50 degrees is pretty darn hot for a plant. So there were a couple of researchers, literally just down the hall from where we were there, who were trying to figure out how these plants could survive under the really extreme conditions. what they found was that these plants could only survive if they had an endophytic fungus in their roots. An endophyte is just inside, so it's inside the plant cells. You have these curvularia that are inside each of those root cells, and you had to have these, again, endophytic fungi in the roots in order for the plants to be able to survive under those extreme conditions. But only if that fungus was infected by a virus. So these are the data, in fact, from that original paper. So these are the happy plants with everything. If you now look at a virus-free plant, it's nowhere near as happy under those kinds of conditions. If it's lacking the fungus, if it's lacking both, just really unhappy. So these are plants that can only grow at high temperatures if they have a fungus and if that fungus is infected by a virus. Now, what is actually going on there? It's a whole area of research that Marilyn Rusink, who's now at Penn State, is very actively trying to research and trying to figure out how the heck this is actually happening. But it's one of my favorite examples of the good viruses, at least as far as the plant is concerned. You know, not very happy at high temperatures, but perfectly happy if it's got the fungus and that fungus has the virus in it. And this is actually called the thermal tolerance virus, the curvularia, which is the species of fungus, and the curvularia thermal tolerance virus. Turns out to be a double-stranded RNA virus, a whole different and interesting story about these kinds of processes. So can we get 100% now on our next clicker question, please? It's a double-stranded RNA virus, actually, which is in these curvularia. Many fungal viruses are that way as well. Now, come on, let's actually get this started so everybody can leave and escape this ridiculously overheated room. Come on, go. Go! Go, 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 go! Do it. There we go. And everyone wants to get out of here. Quick, vote, 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 vote. And if you see another answer that looks better than the first one, vote for it. This is it for class, by the way. If you voted and you're happy with your vote, tension rises. 20 seconds to go. Five, 4, 3. Well, except we had 42 last time. Unless people have come or left, then, you know. Okay, yes, it is E. It's the fungal infection and the fungal virus. Yay. Okay, I will not talk about tobacco mosaic virus, even though it's a fascinating issue. If you're interested, here's the book on tobacco mosaic virus. Again, feel free to come and ask me about that. And I will have office hours now where we will be talking about Lambda. If people want to look at the Lambda video, we can also talk about other questions that people have. Have a good weekend.